Hi, my name is Justin Stewart, and today I'm going to talk about some research for my master's where I looked at the outdoor atmospheric microbiome and saw how it related to the three-dimensional urban landscape in Philadelphia, Pennsylvania, and relate that to indoor transit system air, kind of like a subway. So let's jump in. I chose to do this in an urban area because they're the fastest growing and changing biomes in the world. Approximately 60% of the human population is expected to live in urban areas by 2050. Furthermore, urban sprawl is changing the ecosystem structure and thus its function. Let's look over here at Amman, Jordan, over a 30-year period, for example. We can see that in 1987, there was a very small urban core and there was more vegetation, but as time went on and we go up to 2017, we can see that the urban area has grown exponentially as well as a reduction in vegetation. And all of these changes are subject to biological, physical, and social factors that make up the city. So now that we understand that these urban areas are changing, how are they structured? Essentially, they are composed of extremely heterogeneous three-dimensional landscapes, and they can host distinct functions. And we'll talk about that in a little bit more later on. And while there is a history of studying the urban microbiomes of soil, water, and some air, we are ignoring the three-dimensional aspect of it. So imagine we take our city and we break it up into these 120 square meter blocks or something, and the size can vary. But let's look at just snapshots of the part of the city. This part might be a park where there's grass and water, while over here there's a little bit of pavement and soil. Here we could add in low-rise buildings or mid-rise and high-rise buildings. So a city, this is all found in the same city. This is from New York City. But the landscape is different in different parts of it, as well as the buildings are different also. So just categorizing everything as a building is ignoring a very large part of the vertical urban structure. So we have seen that this does change the ecosystem function. And so for an example on a manuscript I'm also working on that is going off of these urban landscape structure studies called Sterla studies, we are finding that variation in the structure of the urban landscape in New York City is also resulting in the variation in surface temperature in New York City. And so let's think about this at more of a macro scale. Let's put this in a biological context. You ever see a vacant lot in a city? Different plants are going to seed it differently just based off the way that the building is configured. Are you going to find necessarily ones that need more wind dispersal in an area where it's completely blocked off from any wind coming in, or could it be something like animal dispersal? Think of that same kind of concept, but we're applying that to microbes here. All right. So these different microbial ecology studies have largely ignored atmospheric microbes as well as their function and in urban studies. So let's dive right into that. We know that the majority of urban microbiome studies will focus on ecosystems to start ecosystem services such as like nitrogen cycling in the lithosphere or hydrosphere, and then the built environment. But these microorganisms are the main drivers of these biogeochemical processes, as well as influence human health and a bunch of other different theoretical things like niche occupation. So understanding their spatial distribution within the urban landscape becomes critically important. Microbes are in city air. We found that it's previously been seen that the diversity of microbes in the atmosphere does rival that of soil, and that there's quick turnover in community composition. A study I did, also my master's, found that there was high temporal variation in day-to-day samples of the atmospheric microbiome, and that they're largely attributed to a few different things. They're coming from animals, plants, soil, or they're like generalists that can live in a lot of different environments. Although the near surface atmosphere of the atmospheric microbiome has largely been ignored with regards to urban structure. So as you can see here, this is Philadelphia. We don't know what the difference necessarily of the microbes here in these low rise versus the high rise buildings would be versus in, say, a parking lot. So for this, I'm going to move this so everyone can see that better. I drove 600 miles split into 2 different routes around Philadelphia with essentially a tiny vacuum attached to my car that would collect fine particulate matter of 2.5 micrometers and smaller in size. And I split these different routes up. And so I have one that's going to a more vegetated part of the city generally, and that's going to be this route 2. And then one that's a little bit more dense in urban structure, and that's our route 1 here. And within that, we also have subtle variation day to day, and that's due to a couple different things. Sometimes I would stop and get a sandwich, honestly, and I would want to go to a different place. Anyway, these routes were generated to encounter as many urban landscape variations as possible. So that those configurations we talked about earlier, those 120 square meter areas, we wanted to see as many different ones of those as possible. We also collected on 3 subway lines, all on the same on the same day. So we did this 4 times. We would get on these 3 different lines, and we would take samples inside the train car. And it should be noted that 1 car is fully above ground, 1's below ground, and 1 is a hybrid that goes above and below ground in Philadelphia, run on the SEPTA system. And we counted around an average of 30 people on a car at any given time. So what else? After we've collected all of this, let me bring it back actually. We generated our 3-dimensional urban landscape, and you can see that right here where we could see, high rises right here. There's obviously a park over here. There's some water, and then we have just a generally homogeneous area for the rest of the city. We combine that with that, as I was mentioning, a vacuum, that we vacuum these microbes on to these little filters. And then from there, we would take these filters. I'm sorry, did I mention that we drove the car around to collect them? We drove a car with the vacuum attached to it. We would take these filters, I would extract the DNA and then amplify a marker gene sequence, the 16S ribosomal RNA sequence. And then I figured out taxonomy and evolutionary history using the CHIME2 pathway, and predicted the metagenome of these microbial communities using PyCrust2. It doesn't say it there, but it's 2. And shout out to Megan for being super great. That time everything broke down on the side of the highway. So let's talk about who we found. We found 337 amplicon sequence variants across 22 different phyla. No phylum was unique to a single sample or air type, suggesting that all the microbial communities are panmitic or constantly mixing, very vast becking of them. And we found that it was largely dominated by proteobacteria, firmicutes, and actinobacteria. What's really interesting is if you look at this, where on all of these, the last 4 are going to be indoor transit system air, like your subway, and then the other ones are the outdoor atmospheric microbial air. And you can see that they vary with regards to the relative abundances of these taxons, but it is largely proteobacteria in the subway, almost, I think it was around 90%. We also found that they are differing in air by community structure. So here are 2 principal coordinate analyses. Ordinations. And so we're going to see on the left here, we can see that the community structure significantly differed both, it significantly differed between indoor and outdoor air, but it different, and by the outdoor route driven, but it didn't differ in how these are dispersed. And then we find that the same thing is unsurprisingly coming from the functional potential structure. We're finding that outdoor air is different day to day, or from different routes. And we found that indoor air, the indoor transit air and outdoor air, are also significantly different from each other. Although it should be noted that the outdoor airs and indoor transit airs did not differ with regards to how they're dispersed. We also found, unsurprisingly, given our first graph that we looked at, that outdoor air is more diverse and variable, and this was using 4 different metrics. So we have phylogenetic diversity here, where it's much more diverse, and this, although there is one interesting sample in the subway that had very low phylogenetic diversity. Then we also found that they were functionally diverse and differed with regards to that, both by route and, for this one, by route as well as by indoor outdoor, but in this one, they did not differ by route. And then the number of species we found, or the number of ASVs, and then how evenly distributed they are. So we're finding that the outdoor air is more variable with regards to diversity, and it is more diverse than the indoor transit system air. And that does make sense given the fact that you're outdoors and there's wind moving, as opposed to in a train car where it's I hope your doors are closed in the train car, and generally cut off. We also found that they're sourced differently. And again, this all is making sense to each other. We sampled across these diverse landscapes that has water features, green space, unpaved soil, which are likely the sources of the soil taxa that we found. And we found that transit systems are dominated by animals. And you can see that here on this graph right here, and that these sources also differed from each other significantly. But not by route, which again supports the whole story that we're building here. And it's consistent with other studies of both outdoor air as well as transit systems. So specifically, this was in Hong Kong and this was in Colorado. And yes. So am I going to get sick is a really great question everyone asks me when I talk about this. And I'm like, well, I don't know. But we found that of the potentially pathogenic fraction of the different microbes that are in the atmosphere, that the outdoor air hosts fewer but more diverse ones, and that indoor transit air pathogens are largely dominated by gut and, I should say, and skin commensal microorganisms. And that I think it's due likely to the architecture and those sources within that environment can likely explain that variation. It makes sense that to see all of this Enterobacteriaceae in a subway car, because it is filled with humans and it's closed off, and the subway environment in general is just closed off because you have to go underground normally, or except for the ones where we have our outdoor air. So now let's get to the fun stuff. We found that the community structure of the atmospheric microbiome is associated with the variation in 3D urban structure. So our urban structure was not equal throughout the city. And you can see that on this graph here, as well as just a bar graph version of the top, I think this is 10 Sterla classes. And let me just explain these real quick. It'll say TGPL was the most commonly seen one that we had. And you can see that right here with this color, this pinkish color. That means that it was an 120 square meter area. We had trees, grass, pavement, and low-rise buildings, which if you've ever been to Philly or New York City, it's very familiar looking to you. And over here, we can see that the dissimilarity, the Bray-Curtis dissimilarity in the different routes we drove each day is significantly and weakly correlated with the unifrac dissimilarity of the microbes that we encountered on that day. And we did test this with a Mantel test. This is just a visualization of what it essentially looks like, because it's hard to visualize a matrix correlation. Then we also did some univariate statistics, where we found that the richness and functional diversity of the atmospheric microbiome are associated with repeated exposure to specific three-dimensional urban land classes. For example, TGPLM has a significantly positive relationship with functional diversity. So the more often we encountered TGPLM 120 square meter blocks in the city, the more likely we're adding on something, different function that we hadn't previously encountered. And the majority of these relationships were found at the functional level, as well as one that was just at the richness level. I was surprised by the lack of phylogenetic diversity that was significant, although we can see that there are trends that still could be explored further. We did find that functional potential is associated with specific functions are associated with the diversity or the richness of the number of Sterla classes that we saw each day. And we found that there's genes for carbon fixation, sulfate reduction oxidation, nitrogen reduction, and aromatic compound degradation, all suggesting these microbes have the ability to change ecosystem function either on land or in the atmosphere. And what's really interesting is to know that a lot of these were redundant throughout all our samples. And that is good to know that it may be good for ecological resilience under global change scenarios. So let's break down a couple of the different functions we saw. Here I'm just showing metabolic related associations. We found that there were generally more positive associations or correlations as you increase the number of land uses and land classes. But interestingly, there are some downregulations, and they indicating that if you encounter another land class, it doesn't necessarily mean that you're going to be adding other function. It could be taking one away somehow by moving a microbe in or out of the atmosphere. So let's recap. The atmosphere hosts phylogenetically and functionally diverse communities of microbes that co-vary with a 3D urban landscape. Outdoor air is more diverse and is sourced from mostly animals and soil. And subways are likely seeded by its riders and its animal companions, like Tippy, the little dog we saw earlier. Next steps would be probably to do parallel sampling with collecting in different Sterla classes or the same Sterla class throughout the city, and then repeating this in other urban areas. Does anyone have any questions? I do want to appreciate the help from Gerardo Bandera and Jennifer Santoro for analysis, Bradley Reese for sample collection, as well as Kalina Strada's sustainable fashion line, where I took almost all the colors we used for this. And also thank you to the National Science Foundation and Villanova University for the resources. And follow me on Twitter for science jokes and more like yelling about statistics and microbes. Here are my references. I'm gonna move that to the side. I hope you guys have questions. This was a lot of fun. I wish I was with all of you in person. Bye.